Continuation of the previous video. Granulocytes. Granulocytes have irregular shaped nuclei with 2 to 5 lobes and are often called polymorphonuclear leukocytes. Their cytoplasmic matrix has granules that contain reactive substances that kill microorganisms and enhance inflammation. 3 types of granulocytes exist: basophil, eosinophil, and neutrophil. Neutrophils due to irregular shaped nuclei, also called polymorphonuclear neutrophils or PMNs. The neutrophils has a multi-lobed nucleus and a granulated cytoplasm that stains with both acidic and basic dyes. Neutrophils are produced by hematopoiesis in the bone marrow. They are released into the peripheral blood and circulate for 7 to 10 hours before migrating into the tissues, where they have a lifespan of only a few days. Movement of circulating neutrophils into tissues called extravasation. Extravasation takes several steps. The cell first adheres to the vascular endothelium, then penetrates the gap between adjacent endothelial cells, lining the vessel walls, and finally penetrates the vascular basement membrane, moving out into the tissue spaces. Like macrophages, neutrophils are active phagocytic cells. Eosinophils have bilobed nucleus and stains with acidic dye eosin. It comprises 2 to 5% of WBC and are motile phagocytic cells that can migrate from the blood into the tissue space. Their role is important in the defense against protozoan and helping the parasites, mainly by releasing cationic peptides and reactive oxygen intermediates into the extracellular fluid. Basophils have lobed nucleus and stain with basic dye methylene blue. It comprises less than 1% of total WBC. They are non-phagocytic, release substances that cause an allergic response. These molecules include histamine, prostaglandin, serotonin, leukotrienes. Basophils and mast cells possess high affinity receptors for one type of antibody known as IgE. Then mast cells. Mast cell precursors are formed in bone marrow and released into blood in an undifferentiated state until they reach tissues. They have a large number of cytoplasmic granules containing histamine. Mast cells and basophils play a role in allergic response. Then mononuclear phagocytes. The mononuclear phagocytes include monocytes circulating in the blood and macrophages in the tissues. Monocytes are mononuclear phagocytic leukocytes that circulate briefly in the bloodstream before migrating into the tissue, where it becomes macrophages or into dendritic cells. So macrophages are phagocytes derived from blood monocytes. The monocyte is a small spherical cell with few projections, abundant cytoplasm, and many granules. Following migrations of monocytes into the blood into various tissues, they undergo further differentiation into a variety of histologic forms, all of which play a role in phagocytosis, including the following: Kupffer cells in the liver, large cells with many cytoplasmic projections; alveolar macrophages in the lung; splenic macrophages in the white pulp; then peritoneal macrophages, free floating and peritoneal fluid, along with that osteoclasts in bone; then mesenglial cells in the kidney; microglial cells in the central nervous system. First, dendritic cells. Dendritic cells are bone marrow derived cells that descend through the myeloid and lymphoid lineages and are specialized for antigen presentation to helper T cells. Dendritic cells are classified into 4 types: Langerhans cells, interstitial dendritic cells, myeloid dendritic cells, lymphoid dendritic cells. Despite their differences, they all constitutively express high levels of class II MHC molecules. Dendritic cells acquire antigen by phagocytosis. The antigen is processed and mature dendritic cells present it to Th cells. Then follicular dendritic cell, a cell with extensive dendritic extensions that is found in the follicles of lymph nodes. It does not arise in bone marrow and has a different function from the antigen presenting dendritic cells. Although they do not express MHC class II molecules, they are richly endowed with receptors for antibody, which allows the binding of AG-AB complexes. They are of a lineage that is distinct from MHC class II bearing dendritic cells and organs involved in the adaptive immune response. Lymphatic organs. The lymphatic organs are those organs in which B and T lymphocytes maturation, differentiation, and proliferation take place. They are generally divided into two categories: primary and secondary lymphoid organs. The primary central lymphoid organs are those in which the maturation of T and B lymphocytes into antigen recognizing lymphocytes occur. Bone marrow and thymus are examples of primary central lymphoid organs. Thymus gland. The thymus is the site where T lymphocytes mature. Progenitor cells from the bone marrow migrate into the thymus gland where they differentiate into T lymphocytes. It is a flat bilobed organ situated above the heart. Each lobe is surrounded by a capsule and is divided into lobules, which are separated from each other by strands of connective tissue called trabeculae. Each lobule is organized into two compartments, the outer compartment or cortex and the inner compartment or medulla. T lymphocytes mature in the cortex and migrate to the medulla, where they encounter macrophages and dendritic cells. Here, they undergo thymic section, which result in the development of mature functional T cells, which then leave to enter the peripheral blood circulation, through which they are transported to the secondary lymphoid organs. It is in the secondary lymphoid organs where the T cells encounter and respond to foreign antigens. Bursa fabriceus and bone marrow. A primary lymphoid organ was first discovered in birds. In birds, B cells undergo maturation in the bursa fabriceus. This organ, situated near the cloaca, consists of lymphoid centers that contain epithelial cells and lymphocytes. These lymphocytes consist solely of antibody producing B cells. Mammals do not have a bursa fabriceus. B cells differentiate from hematopoietic stem cells in the fetal liver after birth and for the life of the individual. This function moves to the bone marrow, a substructure that is considered to be primary lymphoid organ with functions equivalent to that of the avian bursa fabriceus. Each mature B lymphocyte bears antigen-specific receptors that have a structure and specificity identical to the antibody later synthesized by the B cell. The mature B cells are transported by the circulating blood to the secondary lymphoid organ, where they encounter and respond to foreign antigens. Here you could see a development and action. Hematopoietic tissue, T cell precursor, hematopoietic stem cells, bone marrow lymphocyte, thymus lymphocyte, lymphoid organ, cell mediated immune response, antibody response, T cell and B cell antigen. Here you could see a question. What are the two primary characteristics that distinguish hematopoietic stem cells and progenitor cells? Stem cells are capable of self-renewal and can give rise to more than one cell type, whereas progenitor cells have lost the capacity for self-renewal and are committed to a single cell lineage. Commitment to progenitor cells depends on their acquisition of responsiveness to particular growth factors. Once again, stem cells are capable of self-renewal and can give rise to more than one cell type, whereas progenitor cells have lost the capacity for self-renewal and are committed to the single cell lineage. Commitment of progenitor cells depends on the acquisition of responsiveness to particular growth factors. What effect would removal of the bursa fabriceus have on chickens? The bursa fabriceus in birds is the primary site where B lymphocytes develop. So this removal would result in the lack of circulating B cells and humoral immunity, and it would probably be fatal. Secondary lymphoid organs or tissues. Mature B and T lymphocytes migrate from bone marrow and thymus respectively through the bloodstream to the secondary lymphoid organs. The secondary lymphoid organs are those organs in which antigen-driven proliferation and differentiation take place. The major secondary lymphoid organs are the spleen and lymph nodes and mucosa associated lymphoid tissue or MALT. Spleen and lymph nodes are the highly organized secondary lymphoid organs. The secondary lymphoid organs have two major functions. They are highly efficient in trapping and concentrating foreign substances, and they are the main sites of production of antibodies and the production of antigen-specific T lymphocytes. Spleen. The spleen is the largest of the secondary lymphoid organs. It is highly efficient in trapping and concentrating foreign substances carried in the blood. It is the major organ in the body in which antibodies are synthesized and from which they are released into the circulation. The interior of spleen is a compartmentalized structure. The compartments are of two types, red pulp and white pulp. Red pulp is the site where old and defective RBCs are destroyed and removed, whereas white pulp forms PALs. PALs in the sense PALs, peri-arteriolar lymphoid sheath, which are rich in T cells. The marginal zone located peripheral to the PALs is rich in lymphocyte and macrophage. Approximately 50% of spleen cells are B lymphocytes, 30 to 40% are T lymphocytes. Then lymph nodes. Lymph nodes are small encapsulated bean-shaped structures, normally less than 1 centimeter in diameter, found in various regions throughout the body. The lymph nodes are composed of medulla and a cortex, which is surrounded by a capsule of connective tissue. They are packed with lymphocytes, macrophages, and dendritic cells. The cortical region contains primary lymphoid follicles. After antigenic stimulation, these structures enlarge to form secondary lymphoid follicle with germinal center containing dense populations of lymphocytes, mostly B cells. The deep cortical area of paracortical region contains T cells and dendritic cells. Antigens are brought into these areas by dendritic cells, which present antigen fragments to T cells. The medullary area of the lymph node contains antibody-secreting plasma cells that have traveled from the cortex to the medulla via lymphatic vessels. Lymph nodes are highly efficient in trapping antigen that enters through the afferent lymphatic vessels. In the node, the antigen interacts with the macrophages, T cells and B cells, and that interaction brings about an immune response manifested by generation of antibodies and antigen-specific T cells. Mucus-associated lymphoid tissue. The majority of secondary lymphoid tissue in the human body is located within the lining of respiratory, digestive, and genitourinary tracts. These are collectively called as mucus-associated lymphoid tissue (MALT). There are several types of MALT. Two major MALT include bronchial-associated lymphoid tissue (BALT) and gut-associated lymphoid tissue (GALT). This includes tonsils, adenoids, and specialized regions in the small intestine called Peyer's patches. Then antigens. Adaptive immune response arise as a result of exposure to foreign compounds. The compound that evokes the response is referred to as an antigen, a term initially coined due to the ability of these compounds to cause antibody response to be generated. An antigen is any agent capable of binding specifically to components of the immune response, such as the B cell receptors on B lymphocytes and soluble antibodies. The ability of a compound to bind with antibodies and with the cells of immune system is referred to as antigenicity. There is functional distinction between the term antigen and immunogens. The immunogen is any agent capable of inducing an immune response and is therefore immunogenic. The distinction between the terms is necessary because there are many compounds that are incapable of inducing an immune response, yet they are capable of binding with components of the immune system that have been induced specifically against them. Thus, all immunogens are antigens, but not all antigens are immunogens. The study of antigen-antibody reaction in vitro is called serology. Serological reactions are the basis of all diagnostic immunology tests. Once again, adaptive immune response arises as a result of exposure to foreign compounds. The compound that evokes the response is referred to as antigen, a term initially coined due to the ability of these compounds to cause antibody response to be generated. An antigen is any agent capable of binding specifically to components of the immune response, such as B receptors on B lymphocytes and soluble antibodies. The ability of a compound to bind with antibodies and with cells of immune system is referred to as antigenicity. The ability of a compound to bind with antibodies and with cells of immune system is referred to as antigenity. There are functional distinctions between the term antigen and immunogen. An immunogen is any agent capable of inducing an immune response, and it is therefore immunogenic. The distinction between the terms is necessary because there are many compounds that are capable of inducing an immune response, yet they are capable of binding with components of the immune system that have been induced specifically against them. Thus, all immunogens are antigens, but not all antigens are immunogens. The study of antigen-antibody reaction in vitro is called serology. Serological reactions are the basis for all diagnostic immunology tests. The requirements for immunogenicity. A substance must possess the following characteristics to be immunogenic. Which are they? Foreignness. The most important feature of an immunogen is that its effective immunogen must be foreign with respect to the host. The adaptive immune system recognizes and eliminates only foreign non-self antigens. Self antigens are not recognized, and thus individuals are tolerant to their own self molecule, even though some molecules have the capacity to act as immunogens in other individuals of the same species. Size. The second requirement for being immunogenic is that the compound must have a certain minimal molecular weight. There is a relationship between the size of immunogen and its immunogenicity. In general, some small compounds with a molecular size less than 1000 Dalton, for example penicillin, aspirin, are not immunogenic. Those of molecular weight between 1000 and 6000 Dalton, for example insulin, adrenocorticotropic hormone, may or may not be immunogenic, and those of molecular weight greater than 6000 Da, albumin, tetanus toxin, are generally immunogenic. The most active immunogens tend to have a molecular mass of 100,000 Dalton or more. In short, relatively small substances have decreased immunogenicity, whereas large substances have increased immunogenicity. Then chemical complexity. The third characteristic necessary for a compound to be immunogenic is a certain degree of chemical complexity. For example, homopolymers of amino acids or sugars are seldom good immunogens regardless of their size. Similarly, a homopolymer of poly-alpha D-glutamic acid, the capsular material of Bacillus anthracis, with a molecular weight of 15,000 Dalton, is not immunogenic. The absence of immunogenicity is because these compounds, although of high molecular weight, are not sufficiently chemically complex. Virtually all proteins are immunogenic. Virtually all proteins are immunogenic. Thus, the most common immune response are those to proteins. Furthermore, the greater the degree of complexity of the protein, the more vigorous will be the immune response to that protein. Nucleic acids are poor immunogens by themselves, but they become immunogenic when they are conjugated to protein carriers. Lipids are rarely immunogenic, but an immune response to the lipids may be induced if the lipids are conjugated to protein carriers. Dosage and route of administration. Sufficient dosage of antigen may not stimulate an immune response, either because the amount administered fails to activate enough lymphocytes or because such a dose renders the responding cells unresponsive. Besides the need to administer a threshold amount of antigen to induce an immune response, the number of doses administered also affects the outcome of the immune response generated. The route of administration also affects the outcome of the immunization because this determines which organs and cell populations will be involved in the response. Immunogens can be administered through a number of common routes: intravenous, into a vein; intradermal, into the skin; subcutaneous, beneath the skin; and intramuscular, into the muscle. Antigens administered via the most common route, namely subcutaneously, generally elicit the strongest immune response. This is due to their uptake, processing, and presentation to effector cells by Langerhans cells present in the skin, which are among the most potent APCs. Antigens administered subcutaneously moves first to local lymph nodes. So intravenously administered antigens are carried first to the spleen, where they can either induce immune unresponsiveness or tolerance, and if presented by APCs, generate an immune response. Orally administered antigens, gastrointestinal route, elicit local antibody responses within the intestinal lamina propria. Then haptens. Substance is called haptens from the Greek hapten, which means to grasp. Fail to induce immune response in their native form because of their low molecular weight and their chemical simplicity. Haptens are antigens but not immunogenic. These compounds become immunogenic when they are conjugated to high molecular weight physiochemically complex carriers. The hapten is a compound that by itself is capable of inducing an immune response, but against which an immune response can be induced by immunization with the hapten conjugate to a carrier. So antigen antibody interactions. Next topic, antigen antibody interactions. AG-AB interaction is highly specific and occurs in similar way as bimolecular association of an enzyme substrate. The binding between antigens and immune components involve weak non-covalent interactions. The binding forces are relatively weak and reversible and consist mainly of van der Waals forces, electrostatic forces and hydrophobic forces, all of which require very close proximity between the interacting moieties. The smallest unit of antigen that is capable of binding with antibodies is called an antigenic determinant or epitope. The corresponding area on the AB group combining with epitope is called paratope. The number of epitopes on the surface of an antigen is its valence. The valence determines the number of antibody molecules that can combine with the antigen at one time. If one epitope is present, the antigen is monovalent. Most antigens, however, have more than one copy of the same epitope and are termed polyvalent. An affinity and avidity. The intrinsic association constant. The intrinsic association constant that characterizes non-covalent interaction between single antigen binding site of an antibody with an epitope is termed affinity. Low affinity antibodies bind antigen weakly and tend to dissociate readily, whereas high affinity antibodies bind antigen more tightly and remain bound longer. When the antigen consists of many repeating identical epitopes or when antigens are multivalent, the association between the entire antigen molecule and antibodies depends not only on the affinity between each epitope and its corresponding antibody, but also on the sum of the affinities of all epitopes involved. The term avidity is used to denote the overall binding between antibodies and a covalent antigen or multivalent antigen. So when complex AG having multiple repeating epitopes are mixed with AB having multiple binding sites, the interaction of such type between multivalent AB and AG is called the avidity. Thus, in general, IgM antibodies with 10 antigen binding sites are of high avidity than the IgG antibodies through the binding of each FAB in IgM antibody with ligand may be of the same affinity as that of Fab from IgG. Overall binding between antibodies and multivalent antigen avidity. Then adjuvant. An adjuvant, from Latin word adjuvant means to help, is a substance that when mixed with an immunogen and injected with it enhances the immune response against the immunogen. Adjuvants are often used to boost the immune response in case of weak immunogens or when antigens are available in small amounts. A carrier for hapten and an adjuvant is different. A hapten will become immunogenic when conjugated covalently to a carrier. It will not become immunogenic if mixed with an adjuvant. Thus, an adjuvant enhances the immune response to immunogens but does not confer immunogenicity to haptens. How haptens augment the immune response has remained enigmatic. While many adjuvants have been developed in animal models and tested experimentally in humans, only one has been used for routine vaccination. Currently, aluminum hydroxide and aluminum phosphate are the only adjuvants used for human vaccines. Another commonly used adjuvant is Fruit's complete adjuvant FCA, consisting of heat-killed Mycobacterium tuberculosis or M. butyricum suspended in oil. Then cross-reactivity. Although AB AG reaction is very specific. Sometimes antibody elicited by one antigen can cross-react with an unrelated antigen. An immunologic reaction in which a particular antibody or T cell receptor react with two or more antigens that possesses a common epitope is called a cross reaction. When two compounds cross react immunologically, the compounds will have one or more epitopes in common, and the immune response of one of the compounds will recognize one or more of the same epitope on the other compound and react with it. Another form of cross reactivity is seen when antibodies or cells with specificity to one epitope bind, usually more weakly, or another epitope that is not quite identical but has a structural resemblance to the first epitope. So, antigen presenting cells next, APCs. Before cytotoxic or helper T cells can kill or help their target cells respectively, they must be activated to proliferate and differentiate into effector cells. This activation occurs in peripheral lymphoid organs on the surface of antigen presenting cells that display foreign antigen complexes with MHC proteins on their surface. APCs are any cells that can process and present antigenic peptides in association with class 2 MHC molecule and deliver a co-stimulatory signal necessary for T cell activation. There are three main types of antigen presenting cells in peripheral lymphoid organs that can activate T cell: dendritic cell, macrophages, and B cells. The most potent of these are dendritic cells, whose only known function is to present antigens to T cells. Antigen presenting cells display three types of protein molecules on their surface that have a role in activating a T cell to become an effector cell. MHC proteins, which present foreign antigens to the T cell receptor, co-stimulatory proteins, which bind to complementary receptors on the T cell surface, and T cell adhesion molecules, which enable a T cell to bind to the antigen presenting cell for long enough to become activated. So, receptor for co-stimulatory protein, T cell receptor, foreign particle or peptide, MHC protein, cell adhesion proteins, co-stimulatory protein, T cell and antigen presenting cell. Three types of proteins on the surface of an antigen presenting cell involved in activating a T cell. The invariant polypeptide genes that are stably associated with the T cell receptor are not shown. So, major histocompatibility complex or MHC. Every vertebrate species has MHC genes and products. MHC is a tightly linked cluster of genes, the products of which play a role in intercellular recognition and in discrimination between self and non-self. MHC is located on chromosome 6 in humans and on chromosome 17 in mice and referred to as the HLA, human leukocyte antigen complex, and H2 histocompatibility 2 complex respectively. MHC is highly polymorphic. Many alternative forms of the gene or alleles exist at each locus. Different individuals within the species have different alleles of MHC gene, that is, at the same single MHC locus, different individuals have different types of prototypical gene. The phenomenon of having multiple stable forms of one gene in the population is known as genetic polymorphism. MHC genes are codominantly expressed, that is, every cell that expresses MHC molecule expresses proteins transcribed from both the maternal and paternal chromosomes. Two major sites of MHC genes known as class 1 MHC and class 2 MHC and their cell surface expressed products, MHC molecules, sometimes referred to as MHC antigens, are present. Class 1 MHC gene expresses glycoprotein called MHC molecule, sometimes referred to as MHC antigen, on nearly all nucleated cells. Each MHC class 1 gene codes for a transmembrane glycoprotein of approximate molecular weight 43 kilodalton, which is referred to as alpha or heavy chain. It comprises three extracellular domains, alpha 1, alpha 2, and alpha 3. Every MHC class 1 molecule is expressed at the surface of a cell in non-covalent association with a small invariant polypeptide called beta 2 microglobulin. Beta 2 microglobulin, molecular weight 12 kilodalton, which is coded from another chromosome. Beta 2 has a structure homologous to the single Ig domain. The T cell molecule CD8 binds to the invariant region of all MHC class 1 molecules. Class 2 MHC gene expresses MHC molecule constitutively under all conditions only on APCs, macrophage dendritic cells and B cells. Many other cells such as fibroblasts and endothelial cells and mouse macrophages may be induced to express MHC class 2 molecules by factors such as the cytokine interferon and that are released during the response to infectious agents. In summary, in the absence of inducing factors, most cells express MHC class 1 molecules without expressing MHC class 2 molecules. Certain cells such as B cells constitutively express both MHC class 1 and 2 molecule. By contrast, very few if any cells express MHC class 2 in the absence of MHC class 1. MHC class 2 alpha and beta genes code for chains of approximate number weight. MHC class 2 molecules like MHC class 1 molecule are transmembrane glycoprotein molecules with cytoplasmic tails and extracellular Ig-like domains. Domains are referred to as alpha 1, alpha 2, beta 1, beta 2. MHC class 2 molecules are also members of the Ig superfamily. The T cell molecule CD4 binds to the invariant portion of all MHC class 2 molecules.